Welcome to the Wahl Lab. My name is Razia Naeem, and I'm a research technician here in the lab. One of the most important molecular imaging tools today, and one that we use in the lab, is GFP, green fluorescent protein. GFP was first isolated from the jellyfish Aquaria victoria and exhibits bright green fluorescence when exposed to light in the blue to ultraviolet range. Many colors have now been derived from the green fluorescent protein and red fluorescent protein that was initially derived from coral. One way to use this tool is to use GFP as a reporter gene. GFP can be linked to another gene that produces a protein that you would like to study. In our lab, we use transgenic mice that contain GFP linked to a gene of interest, in our case, SOX10. This allows us to track protein expression. The GFP allows us to track when SOX10 is transcribed and translated by making SOX10 expressing cells green.
Hello everyone, my name is Vikas and today I will be explaining how and why we genotype. Say we inserted a gene for a fluorescent protein into a mouse's genome and breed it with another mouse. How can we tell whether the children's DNA has that gene of interest? To do this, we use a technique called polymerase chain reaction, or PCR, which allows us to amplify short, specific regions of the mouse's DNA by millions of times. We can set this up so that the presence or length of these segments can tell us whether an inserted gene is either present or absent, seen by the pink arrows. The large amounts of DNA can be visualized by staining, but more on that later. PCR works by first denaturing the DNA, or separating the two strands from each other by heat, as seen in the red box. Very small sequences of nucleotides called primers bind to highly specific regions of the DNA strand, and the binding site of these primers determine the region and the length of the segments amplified, as seen in the yellow block. Then polymerase enzymes add free nucleotides from the solution to build complementary strands of DNA, as seen in the orange block and the figure on the right.
Hi, my name is Geoff Wahl. I work at Gene Expression Laboratory, Wahl Lab here. And today I'll be showing you how to isolate DNA from animal tissue and use them to perform polymerase chain reactions to amplify the gene that we're looking for. The first step of genotyping requires an extraction of DNA from our mouse tissue. To do so, we add a lysis buffer that has a detergent to break down lipid layer of the membranes to release the DNA from cells, as well as proteinase K, which is an enzyme that removes proteins that degrade the DNA. This process requires an incubation at 55 degrees overnight. Afterwards, we isolate out DNA using alcohol because DNA is insoluble in the presence of alcohol. Adding isopropanol to the lyse buffer mixture will precipitate out the DNA, as you can see here. Then we clean the DNA precipitate with ethanol to remove any remaining impurities. After evaporating out the alcohol, we resuspend DNA in a buffer that dissolves and protects DNA from degradation. To perform PCR to amplify our gene of interest, we make a master mix with a buffer that provides a suitable chemical environment for the DNA polymerase activity, a nucleotide mixture called DNTPs providing building blocks for DNA synthesis, primers that flank the gene of interest, and a DNA polymerase that has high heat tolerance that can work well under extreme heating condition. Then we add our DNA samples we prepared to provide a source of template for the PCR reaction. This reaction mixture is then moved to a machine called thermocycler, which enables these samples to go through multiple cycles of heating and cooling to actively amplify the gene of interest.
Hi, my name is Cynthia Ramos, and today here at the Gene Expression Lab Wahl, I am going to show you how we visualize the DNA that comes from the PCR reaction by using the agarose gel electrophoresis method.
Agarose powder is dissolved in a buffer solution which serves as a matrix for DNA to flow. A standard is used to compare the sizes of DNA fragments in our samples and is loaded into one of the gels. Next, we pipette some loading buffer and mix it into our PCR reaction. The loading buffer contains a colored dye which allows us to track the DNA as it moves through the gel matrix. It also contains glycerol, which will make the sample heavy so that when we load our samples to the wells in the gel, the sample will sink in the bottom of the well. The tip of the pipette is carefully put into the well and each sample is loaded. The lid is attached and connected to a power supply. DNA is negatively charged, and so all DNA flow towards the positive electrode, and fragments will be separated according to size. Smaller fragments will move faster, and bigger fragments will move slower. The agarose gel contains ethidium bromide, which is a stain that binds to DNA. Once the gel is exposed to UV light, the DNA fragments will give a fluorescent signal, which will enable us to see how far the fragments have migrated through the gel matrix. The size of the fragments that were amplified in our PCR reaction will be compared to the known sizes of fragments in the standard ladder. The image of the gel is captured in a picture.